Hello everyone, I'm Chungman, a second-year PhD student at Gwangju Institute of Science and Technology. Today, I'm excited to present our work that designs a physiology-based computational model for predicting perceptual dissimilarities of users. The growth of tactile information device has led to an increased interest in accurate and effective communication to end users and improve the overall user experience. To accomplish this goal, the tactile signals should be discernible to clearly associate information with them. Usually, pattern designers run a two-step process to find the perceptually well-distinguishable patterns. They carefully select a subset of vibrations in their interest and run a user study to get its dissimilarity matrix using pairwise or cluster-sorted ratings. This process repeats until they find the best patterns that fit to their interest. However, this process has some limitations due to the nature of vibration perception. Humans' pairwise or cluster-sorted ratings are the perceptual responses to a given vibration set, which means that changing one pattern requires running a new user study. Additionally, those ratings require much effort in time and cost, so the vibration pattern design space has been partially investigated so far. We can overcome the limitations if we have a trustworthy metric for the perceptual dissimilarities. However, most metrics for tactile patterns were designed for similarity estimation in specific domains, which implies that existing metrics may not be effective in estimating perceptual dissimilarities. Therefore, we propose a computational model that can predict the perceptual dissimilarities of vibrations. Our model was inspired by physiological knowledge of vibrotactile transmission from the subneural network to the somatosensory cortex. The model was designed using two independent and parallel approaches of neural coding and one-dimensional convolution processes. The neural coding process simulates mechanoreceptors under the skin and the human tactile neural network system. Let's imagine that a vibration is given to the skin. We simulate the propagation of the vibration, which decreases by the square as the distance from the stimulus center increases. We then apply the four-channel theory about characteristics of four types of mechanoreceptors. First, we assume the single-point stimulation, and four mechanoreceptors are co-located at the center location. Next, we simulate the spatial distribution of activated receptors by considering the density of receptors and the amplitude of the propagated vibration. Then, we applied bandpass filtering based on the frequency ranges of receptors and generate a group of neural spikes at each receptor type. At this point, the group of neural spikes are aligned to the spatial distribution of each activated receptor type. Next, we simulated the neural current transfer through excitatory and inhibitory processes of the somatosensory system. Finally, we took the neural spikes aligned at the center neuron of the last layer in each receptor pathway and integrated the four neural spikes with weight. With this process, a vibration set is converted to a predictive rate waveforms, and then we calculate the pairwise root mean square error to create a dissimilarity matrix. In the one-dimensional convolution process, the vibrations are processed by taking their absolute values and applying moving averages to generate envelope feature waveforms. Pairwise dynamic time warping is then used to calculate the dissimilarity matrix. Finally, the two dissimilarity matrices are integrated with weight to generate a final predictive dissimilarity matrix. Our model consisted of two parallel processes, and we set 8 parameters to train it. These parameters included the weight of the four types of mechanoreceptors, the number of excitatory and inhibitory layers, and the weight of the neural coding and one-dimensional convolution processes. We trained these 8 parameters to maximize the Spearman's length correlation between the dissimilarity matrix of each of the 6 vibration sets and each corresponding predictive dissimilarity matrix generated by our model. We tested our model with 12 vibration sets. These sets were designed by 3 different perspectives of the training set, the training parameter, and the stimulation type. We then evaluate the performance of matrix by measuring Spearman's length correlation between the dissimilarity matrix from the usual data and our model's predictions. We also use alienation coefficient to evaluate the structural similarity of derived perceptual species. For stimulation types, we considered 2 scenarios. The first scenario was the ideal stimulation, where the human skin vibrates according to the designer's intended signal. The second scenario was the measured stimulation, where the actual vibration acceleration in the skin were used as input. We validated our model by comparing it with 6 representative metrics using 12 vibration sets. Our model's predictions showed robustness for all vibration sets, as it was able to derive significantly correlated perceptual dissimilarities for all datasets. Furthermore, our model reported the highest correlation on average for both trained and untrained vibration sets. We further validated our model by assessing the structural similarity of the derived perceptual species. Our model's predictions successfully generated perceptual species for all 12 datasets and reported the loss alienation coefficient with an average value of 0.32. In contrast, some metrics resulted in completely different perceptual species or could not generate perceptual species at all. Here, we present the perceptual species of one representative vibration set consisting of 8 vibration patterns with varying modulated frequency. Our model's predictive perceptual dissimilarities show the highest correlation of 0.78, and the derived perceptual space was the most similar with the lowest alienation coefficient of 0.25. If you are interested, you can find other pattern set results in our paper. We believe that our model has a potential to serve as a pilot tool for pattern design due to its flexibility for vibrotactile parental tests, which can decrease the cost and time of the vibration pattern design process and accelerate it. For more details, please refer to our paper or contact me. Thank you so much for listening to my presentation.